Hey, Rose. Good morning, everyone.
Morning.
Are you guys staying warm? Sort of. Are all of you guys in dorms right now? How are your temperatures in the dorm?
That's fine.
If you come over to White Science and you come through the ground floor, or in the stairwell, you'll see that they've put up space heaters to keep the building warmer. Strangely enough, I was surprised when I walked in this morning and saw that. Needless to say, my office is a little on the cold side. So I think one of the things I'm going to do today, which I did a couple of other times during the semester and told you about, is I'm probably going to go home after class simply because my house is warmer than my office, even though I have a space heater right next to me. So during office hours today from 11:15 to 1:35, if you need me, you can just email me, and if we need to, we can set up a Zoom meeting. Does anybody need in the lab today? Okay, then I'm just going to leave the lab unlocked, or leave the lab locked rather, since nobody's planning on coming in. Don't forget, we have lab practical on Thursday. So Thursday morning before lab, don't expect to be able to come in and do any last minute cramming on Thursday morning because it'll be closed because I'll be setting up, I'll be in the room setting up the lab. So that is that. All right. Any questions about the exam last Friday? How did it go? How did it feel?
I thought it was long, but I don't know. I kept getting interrupted, so that could have been why.
Okay, so you thought, well, yeah, I talked to you about that during the exam. Elizabeth, were you about to say something?
I was gonna say I thought it was pretty long too, but I also don't have a Apple Pencil, so it just just took a really long time.
I agree.
Okay.
I had to put a text box in for every single one, so that was a little
Okay.
stressful.
Yeah.
That
Okay.
I didn't finish some of the problems that I could have.
All right. I wish you guys had told me that, because what I'll do next time is rather than putting a PDF version on the Moodle site, this is why I asked you guys to like work out how you needed to do this. I'll put both a PDF version and a Word version. So those of you who don't have Apple Pencils, you can just open up the Word version and type things in rather than messing with putting in stupid text boxes. Yeah, I could have done that on Friday. I just didn't know that you guys didn't have the capacity to do that.
It wouldn't have made a difference for me. Like I have an iPad, and so the Word, like it just doesn't work.
Oh, really?
So like a
Okay.
PDF would have been the option anyway.
Okay. So do you have Word on your iPad, Elizabeth?
I don't know if we were supposed to get it through Joule, but it doesn't work. So
Okay.
I don't know.
You need to email Heath Hayes, and he will fix that for you. It's his job to fix that for you. Guy's name is Heath Hayes. He's in charge of instructional technology. If you're having trouble getting that on your iPad or getting it to work, he should be able to help you out with that. So do that before next exam, and then you should have access to Word on your iPad in a way that will work. So, all right, yeah, I will definitely do that for next exam. My bad. I should have asked about that beforehand. It just didn't cross my mind. Because once you're done in Word, you can save your Word file as a PDF and submit it that way. The other thing is if you submit it as a Word file, that's not that big of a deal. I dealt with this all last semester. It's easy enough for me to convert it to a PDF for grading. So did you guys finish everything though? Even though it took slower? Kiara?
I finished. I just know I didn't finish well on some of them.
Okay, all right. So the other thing is, because you're taking tests in a new way, and I think I mentioned this last week, I'm being less rigid about when you turn it in. So if it takes you a couple more minutes because of the testing environment in order to get it in, I'm not going to not accept the test because it got in technically late. I want you to do well on the test, and if it takes you a couple minutes more because you're taking it digitally, then I'm perfectly okay with that. So, and I thought I made that clear last week, but maybe I didn't make it as clear as I could have. All right. Questions about what we were covering before we took a break to take a test? All right, can you guys see my screen? Okay, so we finished up last time. We were talking about the thermal pits in certain groups of snakes and talking about both the pits themselves and how they're innervated, what section of the brain that they go to, which is a section of the brain that handles visual spatial information. And we also talked about the fact that in addition to the pit, which is a highly evolved sort of structure, there's also evolution, correlated evolution going on with, for example, the circulatory pattern in the head of snakes that possess thermal pits, such that it keeps these thermal pits kind of thermally buffered from the heat of the snake itself, so that when they sense heat, they're really sensing heat from external sources, not being complicated by the heat, the body of the animal themselves. So once again, that's not found in all snakes. It's just found in a reduced number of snakes. The pythons, the boas, and the pit vipers are the groups that have these organs. So now we're going to start kind of romping through the groups of organisms that are in the reptilia. So this is the larger vertebrate phylogeny from your book. You can see that we have the lungfishes and the coelacanths down here. We have the caecilians, then we have the anura and the caudata. So the Lissamphibia is the group of extant amphibians. And then we have the amniotes right here, the development of an amnion. And in the amniota are all of the terrestrial vertebrates, the mammals, the crocodiles, the birds, rhynchocephalians,
Did you change slides? Sorry.
I did change slides.
Because it didn't.
Is it not?
It's just showing the one with the pit, so it's not
All right.
updated.
I will go back. Let's see. So here's phylogeny. Okay, phylogeny. Here we have the fishes that were the precursors, related to the precursors of the tetrapods. Among the tetrapods, we have the caecilians, which are not tetrapods at all. They've secondarily lost their legs. We have the anurans, frogs, caudata, the salamanders. And then we have here the origin of the amniotes, including the mammals, the turtles, the crocodiles, the birds, the rhynchocephalians, the tuataras, and the squamata, which is the lizards and the snakes and the amphisbaenids. So on this phylogeny, you can see higher taxonomy mapped out. And the Lepidosauria is this group, this common stem that includes the tuataras, as well as all of the snakes and lizards and amphisbaenids. And so this is what we'll begin with, and then we'll work our way kind of back evolutionarily to crocodiles, turtles, etc. The reason that I cover Lepidosauria first is mainly because that is where the bulk of reptile diversity is found, outside of the birds. And so lizards and snakes are diverse groups of organisms with a complicated and a more complicated evolutionary history than we used to think. We used to think that the squamata, which is the group of lizards and snakes, basically branched off into one group that is the lizards and one group that is the snakes. We used to call this Sauria, and we used to call this Serpentes. This is no longer the way we understand lizard and snake evolution. And so that's part of what I want to get across to you today. So in the Lepidosauria, there are these four groups: the tuataras, the snakes, lizards, and amphisbaenids. The tuatara represents its own evolutionary branch. It's an ancient group of organisms. There's fossil evidence going back millions and millions of years of this group in New Zealand. And it's also a geographically limited group of organisms, whereas the lizards and snakes have kind of a cosmopolitan distribution around the world. So tuatara, the taxonomy on tuataras is constantly in flux. I'm just going to refer to it as Sphenodon punctatus. Some people consider this to be a single species. Some people consider this to be up to 4 different species. It used to be much broader in distribution in terms of its historical distribution, but now it is only limited to the islands of New Zealand. And it shares lots of characters with the squamata. It has the same type of teeth. It has these shared outpocketings of the cloaca, presumably they're homologous to the hemipenes of squamata. They have akinetic skulls, so they have skulls that have considerably more fusion and are less flexible than, for example, the skulls of snakes, for that matter. And they also have autotomic planes in the tail. That one of the things that you'll see in a lot of lizard species is the ability to voluntarily break their tail as a defensive mechanism, so that as the animal runs away, if a predator grabs the tail breaks off, the animal continues to run away, and the predator is left with this tail flopping in its grasp. When we get some geckos into the lab, so in March, if you look at the daily schedule, you'll see that there's 1 week in March, I think it's the week that includes the 18th. You'll see that we have a Tuesday and a Thursday off for spring break days. That Friday, I will not be in class, so we won't have class on that Friday because I'll be field collecting in Oklahoma and Texas. And after that point, I'll bring back a bunch of geckos. And one of the things that you'll see once we have some geckos in the lab is you'll see that their tails tend to break off quite easily. So we're going to play around with geckos just to give you some experience in some of the methods that we use to measure things like gecko speed. Nothing that you're going to be required to write a lab report or anything else on, just so that you have the experience of doing that. And one of the things that you'll have to do is you'll have to learn to handle the animals in such a way that you're not constantly breaking off their tails, because Zach is going to be measuring a sprint speed as part of his senior thesis project. And when you break off a lizard's tail, that generally slows down their speed. So they have the same autotomic planes that a lot of lizard species do. So this is a tuatara skull. It's a diapsid, as are all of the squamata. It has an upper temporal fenestra and a lower temporal fenestra. We talked about this early on. Large eyes. These are the acrodont teeth that you can see. But this is basically what the skull of it looks like. And there's considerably actually more fusion in the skull of Rhynchocephalians than in the other squamata, but they do share this diapsid skull arrangement. This is what Sphenodon punctatus looks like. So in many ways, it looks very lizard-like. It tends to have a much heavier, stockier body than many of the lizards. The head is also kind of disproportionately large compared to the rest of the body. When I was in graduate school, the Dallas Zoo had finally, after years of attempting, had finally got permission from the New Zealand government to basically put some tuataras on display at the Dallas Zoo. I was a student at the University of Texas at Arlington at the time, and in the science building at UT Arlington, there was a large cold room down in the basement of the building. It's where Butch Brodie kept his salamanders. And so it was this large refrigerator room, very large refrigerator room. And like a week before the tuataras were supposed to arrive, the Dallas Zoo's exhibit, which is refrigerated basically because these things come from a cold climate, their climate system broke down and the temperature spiked in the tuatara exhibit. And so rather than put off the tuataras coming to Dallas, what they did was they arranged to have them housed temporarily at the University of Texas at Arlington in the cold room downstairs. And they were housed there until the exhibit at the zoo got fixed and had been tested adequately before they were then moved over. Well, it turns out that the cold room was adjacent to the lab that my major professor had at UT Arlington, and there was a door between our lab space and the cold room. And so it was kind of neat because we could just go in and gawk at the tuataras whenever we felt like. So as far as I know, they're still at the Dallas Zoo. So if you're ever at the Dallas Zoo, it's one of the few places outside of New Zealand that you can actually view a living tuatara. These are slow moving, largely because they live in a cold environment. They live in burrows, and they're a species of concern. They're an endangered species because of predation from introduced predators that they didn't have to contend with before humans came to the island. So then the other branch of the Lepidosauria, which is the squamata, it includes snakes and the lizards and the amphisbaenians. And recent work shows that snakes and amphisbaenians are all derived from lizards. And so if you look at the phylogeny of the squamata, so here's the base of the squamata, everything here is included in the squamata. You see that Rhynchocephalia is not. This is the tuatara. So all of these families of lizards are considered in the squamata, but then you will see that Serpentes is a monophyletic group within the squamata, and the amphisbaenids are also a monophyletic group within the squamata. So we've traditionally put these three groups These three groups, the Sauria, which are the lizards, the snakes, which are Serpentes, and Amphisbaenia, which is the Amphisbaenians. We have traditionally put these as kind of three equally hierarchical, equal levels within the hierarchy, but through recent phylogenetic evidence, that's not a correct way of viewing these things. These are actually all just members of this broader category, Squamata, and we see that what was at one time considered a suborder of Squamata, the Amphisbaenians, are embedded within the Squamata, and Serpentes are also embedded within Squamata. So we don't really call them these things any longer. They're just all squamates. Some of the squamates are amphisbaenids, some of the squamates are lizards, and some of the squamates are snakes. One of the things that you'll see is that there's some pattern to this phylogeny. So we have the Geckoda down here in the lower left, the label Geckoda, and this includes a monophyletic group of organisms that include all of the geckos. There are essentially a number of different families of geckos. Most gecko families have no eyelid. They have a spectacle, a clear scale that goes over the eye, and they're actually looking through the scale so that they have this unblinking gaze. But there are a couple of families that have eyelids. Also, some of these geckos are more arboreal, and others of these are more terrestrial, and so they vary quite a lot, but they all form a phylogenetic group that we generally refer to as geckos. Then we have the Skinkomorpha, which includes a bunch of, for the most part, let's see, one group of Old World species and then three groups of primarily New World species, although skinks also occur in the Old World as well. But the Skinkomorpha includes the skinks, the night lizards, the alligator lizards, and the chordilids, which are the girdle-tailed lizards. And we'll talk about those in some detail. Lacertoidea includes the Teiids, which are the North American race runners, and their Old World equivalents, the Lacertidae. And then you also have embedded within the Lacertoidea, you have the Amphisbaenids. The Amphisbaenids we're not going to talk about too much. They are a not particularly speciose group of organisms, and very few of them occur in the United States, but they are kind of bizarre looking. If you look at the Handbook of Lizards that is up in lab, which is kind of your template for that I've put up there for how to build a dichotomous key. If you go to the very beginning of the lizard dichotomous key, the first lizard group that they split out in that key are the Amphisbaenids, because they lack hind limbs and generally have only 4 limbs for lizards. Then we have the Toxicophora, and the Toxicophora includes the snakes and all of the other lizard groups. So this is by far the most diverse subgrouping within the Squamata. Toxicophora is kind of called Toxicophora because this is also the group that contains all of the venomous groups in the Squamata. So we have Serpentes, which has both non-venomous and venomous representatives in it. But then we have the Helodermatidae, which is on this monophyletic group, including the Xenosaurids and the Anguids. The Hellbenders, sorry, not the Hellbenders, the Helodermatids rather are the Gila monster and the Mexican beaded lizards, and these are also venomous. So you can break up the Toxicophora into kind of 3 main groups: the Iguania, the Anguomorpha, and the Serpentes. The Serpentes includes all of the snakes, and they go in their own phylogeny that we'll see here in a few minutes. Groups that you need to be aware of within this are the Helodermatids, which don't occur in Missouri, but they occur in the southwestern United States. Anguids, which include things like alligator lizards and slender glass lizards, which we don't have in our collection, but they do occur close enough to Missouri that we need to be paying attention to them. We're going to talk about the varanids simply because they're a group of large-bodied species that are, in some cases, top predators in certain environments in the world. But we're mainly going to talk about in class the Crotophytidae, the Dactyloidea, the Iguanidae, the Phrynosomatidea, and the Polycrotidae. These are all families that we have here in Missouri and in the southeastern United States. And we're going to ignore, for the most part, old world species like agamids and chameleons, Chameleonidae. But anyway, the main thing that I want you to take from this particular group is that Rhynchocephalians are distinct from all of the other squamates. Squamates, though, have monophyletic groups that are oftentimes indicative of these large groups like geckos that we think of as being tightly related to one another. But then embedded within the squamates are amphisbaenids, which are also distinctive, but they're not distinctive in terms of their own independent evolutionary branch within the squamates. They're embedded within the squamates, and then the serpentes, the snakes, are also embedded within the squamates. So just a much more complicated way of thinking about the evolutionary relationships of these three groups than we used to think of them as. Questions about the phylogeny? All right, so one of the characteristics that unites the squamata are the presence of hemipenes. Hemipenes are these paired copulatory organs that are in the male. They're stored at the base of the tail. So when you go to sex a lizard or a snake for that matter, there are a couple of ways of doing this. One way of doing this is to just look at the base of the tail. Oftentimes on males, the base of the tail will be thicker because it houses these hemipenes that are stored in the base of the tail. This is the cloaca, so this is the anterior end of the lizard. This is the posterior end of the lizard. Another way of doing this is you can take something like a bobby pin and you can probe into the cloaca. And when you probe into the cloaca, if you direct your bobby pin to the posterior end, you will feel this inverted sac, this opening that goes back. And females also have that opening as well, but the male hemipenis is much longer, and so the degree to which you can insert a bobby pin or an actual reptile probe, they actually make specialized reptile probes for this purpose, but there's no reason that a bobby pin doesn't work. And if you can insert it deeply into the tail, that's indicative of the fact that there are hemipenes there. And if you can't insert it deeply into the base of the tail, that's indicative that you have a female. Now you can't force it in there because you will damage the animal, but if it doesn't take long to get a feel for where the hemipenes are. The other thing you can do is you can just take your thumb and press your thumb at the base of the tail, and when you press your thumb at the base of the tail, you can force the animal to evert the hemipenes. And so there are three different ways in which you can test whether or not whether you have a male or a female. I generally try and do this without forcing the animal to evert the hemipenes or without using a probe. Oftentimes without much training at all, without much experience at all, you can just look at the tail of the animal and ascertain whether it includes hemipenes or not. They're forked, so hemipene refers to half penis, so you have a half penis over here and a half penis over here. They're bound together at the bottom. Oftentimes you will see that they have hooks and barbs on them. Oh, and I thought I had some photos of actual hemipenes. But they can take on all sorts of different shapes, and in many cases, the shapes of the hemipenes are actually useful in terms of taxonomy and phylogenetics. And so groups will have shared characteristics that are related to hemipenal structure. Acrodont teeth basically are teeth where you have the tooth sitting on top of the jawbone like this. Pleurodont and modified pleurodont teeth are teeth that are located on the inner face of the jawbone rather than sitting on top of the jawbone. And in the case of the squamata, all of the squamata have acrodont style teeth. So reproduction in sex is very complicated in the squamata. And the reason is that if it can be found in the animal world, chances are there is a snake or lizard that doesn't. So most species are oviparous, meaning that they lay eggs, but about 20% of the species are viviparous, and viviparity in the snake and lizard groups is just due to a bunch of convergent evolutionary events. So there isn't one synapomorphy of viviparity, and then everybody who evolved from that common ancestor is viviparous. You have over 100 independent evolutionary derivations of viviparity. And as it turns out, a lot of the viviparity that we see in lizards and snakes is actually related to elevation. Low elevation species are generally oviparous, but as you look at higher elevation occupying species, you see that viviparity is much more common if you live at higher elevations rather than low elevations. Of these viviparous species, all of them have at least some form of a rudimentary placenta such that the female is actually transferring nutrients and energy to the developing embryo, as opposed to what we used to refer to as ovoviviparity, where there's just an egg that is developing inside of the female and doesn't receive any provisioning from the female beyond the yolking of the egg. Skinks, in some cases, have well-developed placenta, and when you look at RNA sequencing, if you do an RNA-seq study of the placenta of skinks, you'll see that many of the same genes that are activated in skinks are also the same genes that are activated in mammals that have placentas. So even though these are evolutionarily independent developments of placental reproduction, they're relying on many of the same genes, which is kind of cool from an evolutionary standpoint. Plenty of these groups experience parthenogenesis. Do you guys know what parthenogenesis is?
I think it's when the ovum develops without fertilization.
Yep.
So it's like
that's
only
exactly.
female species.
Yep. So these would be all female species, and they basically reproduce by prompting the unfertilized ova to go through a mitotic division without cytokinesis, and that's how they get the development of a new individual. And these are the families where you find parthenogenesis. So the Agamids are an Old World group. Geckonidae are both Old World and New World geckos. Teiidae are New World race runners. Gymnothalmidae are Old World. Lacertidae are Old World. Xanthuciidae are New World. And in all of these cases, the parthenogenesis is due to hybridization. So not all of the agamids are parthenogenic. Not all of the geckos are parthenogenic. Not all of the race runners are parthenogenic. But what happens is when two closely related species hybridize with one another, oftentimes the resulting offspring end up being parthenogenic hybrid. And this is happened a number of times in the desert Southwest. So you will see in the Western field guide, which you guys have the Eastern field guide, there are a number of Teiid species, these race runners. We have one species in Missouri, the six-lined race runner, that occurs down in the Ozarks, the Ozark Plateau region, especially in the southern part of the Ozark Plateau. It is a non-parthenogenic species, but it has relatives throughout the desert Southwest. And so in the Western Field Guide, there are a number of unisexual species as well as all of the bisexual species that we have within the TIDs. So hybridization occurs when two species hybridize with one another. They produce a daughter species that can reproduce through parthenogenesis and is not reproductively compatible with either of the parent species. And so it's a case of speciation by hybridization also. And then the other thing that we have to think about when we think about the squamata and reproduction is temperature-dependent sex determination. So that's what TSD stands for, temperature-dependent sex determination. Who can describe for me what temperature-dependent sex determination is about? Quinn, did you just make a noise?
Yeah, I think it has something to do with like where the egg is laid and like depending, I can't remember which one, but like whatever like temperature it's at, like if it's at a higher temperature, then it's, I can't remember what it is, but it's either male or female. And then if it's a lower temperature, it's going to be like the opposite sex. I can't
Yeah,
remember.
and the reason that it's hard to remember is it varies by species. And so in some cases, the warm incubation temperatures lead to females and cold leads to males. In some cases, warm leads to males and cold leads to females. In some cases, females are produced at both warm temperatures and cold temperatures, and males are produced at intermediate temperatures. But it's the general idea that the temperature at which the egg is incubated, and especially for a particular window of time during incubation, those temperatures then determine what the sex of the offspring will be. So rather than the sex being determined by chromosome, as it is in humans, where you have an XY and an XX, indicating male and female from a chromosomal perspective, these don't have sex chromosomes as much as they have genes that are turned on and off as a result of environmental stimuli like temperature, and then those genes being turned on and off at particular times leads to differences in hormone levels, which then lead to differences in the sex. So temperature-dependent sex determination is not limited to the squamata. It's also found in turtles and crocodilians, but it's found in a number of different families, although it's the exception. Pardon me, it's the exception rather than the rule. Tail autotomy is also a characteristic that is pretty common. We've already talked about it. It occurs in both snakes and lizards, and there are two different kinds of autotomy. One type of autotomy is under voluntary control, and another type of autotomy is involuntary. The intravertebral, so when you look at a vertebra, you have the centrum, you generally have some anterior processes and some posterior processes, and then you have usually a spine coming off the dorsal surface of a vertebra, and then these spines articulate with spines on the anterior and posterior end of the vertebra that they're next to. But one type of autotomy, there's basically a cleavage plane that goes down the middle of the vertebra. And so when autotomy occurs in this group, the intravertebral group, it actually breaks a vertebra in half, up and down. And then the tail is lost. And in that case, the tail will then regenerate. Intervertebral basically involves the breaking off of the tail between the vertebra. So there is no cleavage plane within the vertebra. And in organisms that do this type of tail autotomy, the tail does not regenerate. In geckos, which is the group that you will work on a little bit in lab, they have intravertebral autotomy, and the tail regenerates, but the regeneration is different from the regeneration in salamanders. So we talked about tail autotomy in salamanders also. Lots of salamanders, well, virtually all salamanders can autotomize their tail, but when they autotomize their tail grows back, and the vertebral column basically looks just like the vertebral column that was originally there. In lizards and snakes, the vertebral column doesn't grow back. What grows back is basically a notochord, a connective tissue structure that serves as the base of the tail, but the vertebral column, the vertebral structure doesn't tend to grow back like it does in salamanders. And then there are some groups that do not autotomize their tail at all. So chameleons have a prehensile tail, and they use it in locomotion a lot, and probably because of that, their tail does not autotomize. The heliodermatids have a very thick tail that is used for energy storage, and once again, probably because of that, they tend to not autotomize their tail. Lanthanodidae and Varanidae are just two types of varanid lizards, or varanid-like lizards. They tend to have very stout tails and also don't autotomize. And in some skinks, they use their tail for locomotion quite a lot and will tend not to autotomize. Yeah, so once again, this is showing you vertebra with fracture planes. The fracture plane is where the break actually occurs. Once that break has occurred, it starts growing this cartilaginous rod, and that cartilaginous rod essentially serves as the kind of skeletal support of the tail. This is actually a Mediterranean gecko. This is the species of gecko that Zach is working on in lab. This is a Mediterranean gecko who has lost its tail at the very base of the tail. This is what a natural original tail looks like. I guess it's not really natural versus not natural. This is an original tail. You can see that it has the same types of scale patterns and things that are found on the body. It has the same type of coloration that is found on the body. When that tail grows back, though, you can see that there is definitely a break point right here. This is the regenerated tail. It generally has a totally different scale type on it than what was on the original tail. It oftentimes will lack pigmentation, and it also lacks the flexibility that the original tail had. But once again, it can be autotomized again right there at that point, so that it can continue to serve as an anti-predator mechanism. So tail autotomy. And if you work with geckos for very long at all, you will occasionally break tails. That is just the consequence of working with geckos. They're very, very fragile. The slender glass lizard, which is an anguid here in the United States, is called a glass lizard because it's one of the legless lizards. And one of the things that it is prone to is its tail breaks off really, really easily. So that's the whole reason that they're called glass lizard, is because of their tail breaking off. Perfectly natural thing to happen to a lizard. There's plenty of limb reduction in the squamata. Sometimes it can just be a couple of phalanges that are lost, but limblessness has evolved lots of different times independently. It is most common in skinks. All of the snakes obviously have lost their limbs, and then the glass lizards, as I was just talking about, the legless lizards, the anguidae, are also in a group that has lost their limbs. So here are examples of of limb reduction. So this is actually all within the skinks. This is all within a single genus of skinks, actually. So here you have kind of a normal looking skink in the upper left-hand corner, which has all 5 digits on the forward limb. And then when you look at other species, they have lost digits, they've lost entire fingers, or fingers have been reduced to just a single phalange in some cases, until you finally end up with legless representatives that have lost all but the most rudimentary parts of the wrist, essentially. And so skinks are one group in which you can have everything from fully limbed organisms to organisms with little reduced limbs, reduced hind limb here, reduced hind limb here, to the point that you don't have any evidence on the outside of hind limbs in other species. And this is all variation within a single genus. This is, if I recall correctly, this is a genus of Australian skinks. So since we're talking about the Toxicophora, which is the group that the squamates are in, we need to talk about venom and venom delivering structures. And so there are, venoms in general are complex mixtures of proteins. So there are genes for venom production in the genome of these organisms, because all of these venoms are protein based. But venoms have been very difficult for people to study because it isn't just one particular compound, it's actually a soupy mixture of lots of different compounds that make up a particular venom. Within that, though, we can define 2 different kind of classes of venom. One type of venom is referred to as hemolytic, the other venom is referred to as neurotoxic. Hemolytic venoms are the type of venoms that you generally find in things like rattlesnakes and the other vipers. These are venoms that are composed largely of basically proteins that are related to digestive enzymes. These are enzymes that are going to break down tissue. And so they're called hemolytic because they basically lyse cells. They break down the structural integrity of cells, and they will break down the structural integrity of blood cells. And this is actually one of the dangers of hemolytic toxins, is they're very good at destroying blood cells. And so they cause a lot of internal bleeding, but then they also can cause plenty of other types of tissue damage. As I mentioned, hemolytic toxins are mainly found in vipers and pit vipers. Neurotoxic venoms are venoms that don't necessarily do much tissue damage, but they function primarily by blocking neurotransmitters, so blocking the transmission of nerve impulses. And this is then limited to a number of families. The helodromatid lizards, which are the Gila monster and the Mexican beetle lizards, have neurotoxic venoms. The varanid lizards have neurotoxic venoms. Some viperids produce neurotoxic venoms, but then all of the venomous species in the Elapidae produce neurotoxins, and some members of the Colubridae. So now we have to talk about the Colubridae. The Colubridae is a family of snakes that used to be just kind of the trash can of the taxonomic trash can of snakes. If you had a snake and it was a generalized sort of snake and it didn't belong to any of the other families that are more distinctive, species just got kind of thrown into the Colubridae. Since then, and with the rise of molecular-based methods in taxonomy, the Colubridae is being split up into a number of different families. Most of the species in the Colubridae are not venomous, but when they are venomous, they can sometimes be some of the most venomous species on the planet. And so, for example, one colubrid from Africa that is very well known for being very dangerous snake is the boomslang. It produces a neurotoxic venom, but unfortunately, it looks just like any other colubrid that you would see, which are generally harmless. When we are in the field and we see a snake, do not pick up the snake until we're absolutely sure what the identity of the snake is. I don't want you to be afraid of snakes because there are actually relatively few snakes in Missouri that are venomous, and the ones that are venomous are relatively uncommon. But when we're in the field, I do not want you to get bit by a venomous snake. So your first response when you see a snake should be to leave it alone and don't attempt to collect it or handle it until you're absolutely sure of what the identity of the snake is. I flipped a rock at Konza Prairie out in Kansas a number of years ago, and under the rock was a copperhead, and I didn't realize that it was copperhead until I lifted the rock and kind of poked my head up over the rock, and then as soon as I saw it, I just let the rock fall back because I did not want to be any part of that. And I was in the field collecting a couple of years ago, and I was actually digging through the grass looking for something else, and I saw, just more with my subconscious than with my conscious, I saw a snake pattern. And the first thing that I did was I withdrew because I saw a snake, but it was down in the grass and I couldn't tell what kind of snake it was based on the little bitty piece of color pattern that I had available to me. I picked up a stick and I pulled back the grass until I could see enough of the body of the snake that I could tell that it was actually a prairie king snake, at which point I reached down and picked up the snake and looked at it. But I myself am cautious around snakes. If I can only see a little section of a snake and I'm not exactly sure what it is, I will generally take precautions until I've ascertained what its identity really is. And at that point, I'll then decide what I'm going to do in terms of dealing with it. When we talk about snakes in lab, you'll see that the venomous snakes that we have here in Missouri are relatively easy to identify. But until you've gone through that training, better to just leave snakes alone. Venomous snakes should always be respected. There's was a herpetologist at the Los Angeles County Museum who did work on snakes in Southeast Asia, and he had snakes in pillowcases, which is oftentimes how you transport snakes. He reached his hand inside of a bag that had a venomous snake in it. It bit him, and he died. And he no longer works at the Los Angeles County Museum because he is dead. So, treat them with respect. Don't ever reach into a bag unless you are absolutely sure you know what is in the bag. And oftentimes it's just better to dump the snake out of the bag and then deal with the snake out of the bag rather than in a bag so that you can actually see the snake. Zoos oftentimes have large collections of venomous snakes, and so they will keep on hand antivenin. Antivenin are protein compounds, once again, that are basically antibodies to venin. And when you get bit by a snake, you can inject antivenin, and it will counteract the effects of the snake bite, and oftentimes save your life and sometimes prevent tissue damage and things like that. One of the problems with snake bites in the world is that pharmaceutical companies, which are generally the people who produce antivenins, are interested in making profit, and unfortunately, antivenins are very difficult to produce and expensive to produce, and they get produced for a very small market. And so many of the pharmaceutical companies that used to produce antivenins no longer produce them. And so there's actually a shortage of antivenins globally, which is problematic, not only for people who work with venomous snakes and things like zoos, but is also problematic for people who just live in places where there are a lot of venomous snakes. Because venoms are complex, the evolution of venom production is also complicated, and there are lots of questions out there about the evolution of venoms and their constituencies. If you are looking for an area of herpetology that has biomedical importance and also is still kind of a wide open field, the biology and ecology and medical applications of venoms is actually one area that is still largely unexplored in herpetology. So here we are back at the phylogeny. I have no idea why the phylogeny is there. Oh, except that we're going to talk about, go back and revisit temperature-dependent sex determination. So before we move into that, do you have any questions about anything we've covered up to this point? I feel like I've been talking a lot now. Okay. So we've talked about sex determination, but we haven't talked about it in detail. And Elizabeth Quinn mentioned that she can't remember which way things go, and the reason that it's hard to remember is because it's a complicated deal. So what I want to do is I want to talk in some detail about sex determining mechanisms. So what we typically have when we go through meiosis is you start off with a cell that hasn't gone through the S phase. When it goes through the S phase, what it does during the S phase is it duplicates the chromatids on the chromosome. So you go from chromosomes that have one copy of the DNA to chromosomes that have two copies of the DNA. During prophase of meiosis, you have synapsis occurring. Somebody remind me what synapsis is. Reach back into your genetics class. Kelsey,
When
what
the
is synapsis?
chromosomes pair- When
What's
the
that?
chromosomes pair,
Yeah, so these are homologous chromosomes. So remind me what a homologous pair of chromosomes is.
like they're the same chromosome, but they don't. So like the same number.
Okay,
They go
so
together,
we
but
have
they have the traits.
what's
right?
that?
They're the same, but they don't necessarily have like the same copy of the trait.
Okay, so yeah, so we have 23 pairs of chromosomes. Each of those pairs is a different homologue. One of these pairs you got from one parent, the other you got from the other parent. So even though the gene for a particular trait may be located here on this chromosome and here on this chromosome, they don't necessarily have the same allele for that trait on the two chromosomes. At synapsis during prophase 1 of meiosis, you basically have the homologous chromosomes pairing up. And while they're paired up, what can they do?
Crossover.
Yeah, they have crossing over, which is just the process by which the ends of the chromosomes will break off and become reattached to the other chromosome. And as a result of this, you get more genetic diversity in your gametes that you're producing. In this case, you're producing an egg and 3 polar bodies because this is a female going through meiosis. And it's just this egg right here, which will be the one that is capable of being fertilized. And so you can see that this chromosome is different from this chromosome, which is different from this one and different from this one. And so this is what typically happens in meiosis. You start off with one diploid parent cell, and you end up with 4 haploid daughter cells. Each of those daughter cells is genetically different from one another. And so there are other ways in which meiosis can occur. And in some cases, this is what we call endoreplication. You basically get, let me think, what am I? Oh yeah. So in this case, what you have is you have 2 chromosomes where you have the 2 homologs pairing up with one another in ways that you would expect, so that when you get crossing over occur, you get this genetic mixture of chromosomes in the end. Each of the chromosome complements of each of these 4 chromosomes is genetically unique. But you can also get situations where the sister chromosomes pair with one another such that anytime any crossing over occurs, you don't get any genetic increase in genetic variation because you haven't had homologs pairing with one another. You actually had during this replication process, you've created new chromosomes. So instead of replicating, so the reason it's called endoreplication, instead of just replicating a single chromosome, you've actually replicated 2 copies of the chromosome. So now you have more chromosomes than you actually need. Normally, you would see homologs pairing up such that the homologs are different from one another in terms of their genes. But occasionally you can have sister chromosomes pairing when you've created too many copies of them. And then that tends to reduce the genetic variability of the daughter cells that you have. This is a modification of kind of what we normally think of meiosis occurring. So when we think about the general ways in which we produce gametes, there's more variation than you probably learned about in your genetics class. And we'll just break there because when we get into temperature-dependent sex determination, it's kind of an involved sort of process. So we'll just cover that on Wednesday. Expect an email from me between now and Wednesday about some family assignments, because after we finish this, we're going to be getting into the actual different groups. And so I'll assign some taxa just like we've talked about, just like we did with the frogs. And that will be that. Questions?
I have a question about the lab practical.
Yeah.
Do we have to know the authority of the species?
No, that would be crazy.
Okay.
Yeah, it's provided on the list just for completeness, but no, that would be too nutty.
Okay, thank you.
Other questions?
I also have a question about the practical. I don't know if I just like misheard this on Thursday, but did you say, so we like just have to know the ones that we've looked at in lab? Because you said something about like there being like 20 different species that we would have to know for the practical, but there's only like 12 or 13.
No, I can't ask you about things that you haven't looked at in lab. No.
Okay.
So that was a rough pull it out
Okay.
out of thin air estimate on how many species are out on the table.
Okay, got it.
Yep, just what's in lab. Other questions? How are you guys feeling in terms of preparation for Thursday?
I feel pretty good about it.
Okay. All right. Well, as always, let me know if you need into the lab, and I will unlock the lab for you. All right. I will see you on Wednesday.
Can you let me and Rose into the lab tomorrow at 9?
I can. Yeah, sure. Let me
That'd
make
be great,
myself a note. Done. I've got a
Thank
note
you.
reminding me. All right. See ya.